Pouring culture media in the laminar flow in the front of burner. Burner actually kill the microbes, so it is very necessary that the pouring must be done near the flames. It is very easy. Do it one by one, assuring that no microbes were there in your pouring media, so that it can be easily poured one by one. Properly sanitize the hands with alcohol and place the plates to other side when it is poured. Take some new slides, new petri dishes, new culture dishes, and pour them one by one. All plates are autoclaved and sterilized. It is very necessary to autoclave and sterilize before pouring. Otherwise, it may be contaminated. It may become contaminated, and this contamination cause the failure of the future prospects, that is the experiments. So to avoid the failure of the experiment, sterilize the plates, and pouring also done with very careful